You know, deep inside every single one of your cells, there are these tiny, powerful motors constantly at work. This is the story of how scientists tried to weaponize them against cancer, a story of incredible precision, an unexpected plot twist, and the brilliant science that came out of it all. So it all starts with a question that unfortunately is really familiar to a lot of people. We all know chemotherapy can be a lifesaver, but it's also famously brutal on the body. We're talking hair loss, nausea, nerve damage. Why is that? The answer to that question is actually the key to the whole breakthrough we're going to explore today. See, the reason for all those side effects is that for a long, long time, our main weapon against cancer has been, well, let's just say it's not exactly a scalpel. It's more like a sledgehammer. Powerful, for sure, but not very precise. Okay, so look at the comparison here. On one hand, you have the old approach, traditional chemo. Think of it like a bomb. It attacks this building block called tubulin that's in all our cells, the healthy ones and the cancerous ones, and that causes massive collateral damage. But on the other hand, a new idea started to form, a precision strike. What if instead we could target something that's only really active in rapidly dividing cancer cells? That was the goal, a smarter weapon that hits the target and only the target.
So let's meet the heroes of our story. These are the tiny engines that scientists were betting on to create a much smarter way to fight cancer.
So what are kinesins? Well, they're pretty much exactly what they sound like, microscopic motors. These are actual proteins that literally walk along these tiny tracks inside our cells, called microtubules. They're always busy, carrying cargo, building structures, all powered by ATP, which is basically the cell's universal energy currency. And we're not just talking about one or two of these things. This is a massive superfamily of proteins. Scientists have found over 650 different types of them across various life forms. And right now, there are about 45 different kinds working inside your body. That huge diversity just tells you how absolutely vital they are. Okay, so what are all these little motors doing? Well, they have two main gigs. One is basically being a microscopic postal service. They transport materials all around the cell. But the other one, and this is the one that's crucial for our story, is their job in powering cell division, also known as mitosis. And that is what really got the attention of cancer researchers. All right, so with 45 different motors to choose from in the human body, scientists had to pick a specific target. And they found a frontrunner, a particular motor that seemed like the perfect weak spot in a cancer cell's armor. And that frontrunner was a kinesin called EG5. Its job is absolutely essential for one very specific task, building something called the mitotic spindle. This is the incredibly complex machine a cell has to build to perfectly separate its chromosomes right before it splits into two new cells. The theory was, if you can stop EG5, the whole process should just grind to a halt. So how does this actually work? Well, imagine a cell has these two little organizing centers. When it's time to divide, the EG5 motors get right in between them and they start to push. They act like tiny little jackhammers, forcing them apart. This pushing action is what creates that stable, two-poled spindle, the scaffolding that's absolutely essential for sorting chromosomes correctly. Now here's the brilliant part of the strategy. You introduce a drug that blocks EG5, and it's like you jam the motor. Suddenly that pushing force is gone. The two poles just collapse back on each other, and you get this chaotic, single-poled starburst shape. This thing is called a monoastral spindle, and for a cell, it is a catastrophic failure. It can't divide, so it triggers its own self-destruct sequence. Game over for the cancer cell. And you know what? The early results were spectacular. In the lab, in petri dishes, and in mice, this strategy worked unbelievably well. These drugs were so effective, they were causing the total regression, the complete disappearance of human tumors that had been grown in mice. The hype was absolutely real. It really seemed like a new, targeted, less toxic cancer therapy was right around the corner. But of course, science is rarely that simple. When this incredibly promising new drug moved out of the lab and into human clinical trials, the story took a sharp and pretty disappointing turn. And here is that twist. In human patients, the results were just stunningly different. Just look at the chart. Complete responses, as in tumors completely disappearing, 0%. Partial responses were incredibly rare. For most patients, the absolute best outcome they could hope for was stable disease, which just means the tumors stopped growing for a while. The magic bullet had completely missed the mark. Now, there was one silver lining in all of this. That original prediction about side effects, it turned out to be correct. Because EG5 is mostly active during cell division and not so much in, say, our nerve cells, patients didn't get that awful neurotoxicity that's so common with older chemo drugs. So the idea of targeting worked, but the effectiveness just wasn't there.
So this led to the central mystery, right? Why would a drug that completely obliterates tumors in mice barely make a dent in humans? Suddenly, scientists had to become detectives and figure out what was going on. So their investigation led to three main theories. First off, speed. The tumors grown in labs and mice divide incredibly fast, which gives the drug a ton of targets, but a lot of human tumors actually divide much more slowly. Second, it turns out cells have a backup plan. There's another motor, a kininin called KIF15, that can actually step in and do EG5's job if it gets blocked. The cancer cell had a workaround. And third, it's possible that the first-generation drugs just weren't powerful enough for the job in humans. Now, this could have easily been the end of the story, a failed drug, a dead end. But in science, a failure is often just a stepping stone to a much deeper understanding. The lessons they learned from EG5 paved the way for brand new, even smarter strategies.
So the research didn't stop, it just got a whole lot more sophisticated. Instead of giving up on kinesins entirely, scientists went back to the drawing board, this time armed with all this new knowledge about how tumors really work. And this led them to some really exciting new targets. One is a kinesin called CENPE, which helps align the chromosomes. But maybe the most clever target of all is a kinesin called HSET. And this is where it gets really cool. See, HSET is not essential in our normal healthy cells, but some really aggressive cancers develop extra organizing centers. It's a flaw that should kill them. These cancer cells, though, have evolved to use HSET as a special tool to bundle those extra centers together, which is what allows them to survive. And that is the absolute essence of this new strategy. Targeting HSET is like removing a very specific tool that only these very specific cancer cells are using to stay alive. Normal cells don't need it, so they should be completely unharmed. But for these particular cancer cells, taking away that tool is catastrophic. It is the very definition of a targeted therapy. And that really leaves us with a final thought. This journey from the sledgehammer approach of early chemo to the hyper-specific targeting of something like HSET tells a really powerful story. Maybe the future of cancer therapy isn't about finding one single magic bullet that works for everyone. Maybe it's about understanding the unique biology of each individual tumor so we can find the one specific key that will unlock its destruction.